Hello. Sorry about that. There's some technical difficulties on our side over here. But Ilhan, if you're still there, I want to thank you for your presentation. That was really fantastic. Moving on, we're going to move on to Maya as the next presenter. Oh, never mind. Okay, Erin, sorry, I didn't see you there. So, okay, I'm so happy that you're here. So I'm just going to let you go and present when you're ready.
Can you hear me okay?
Yeah.
Okay, awesome. Hi everyone, my name is Erin Richardson, and my goal is to one day become an astronaut for the Canadian Space Agency. As you know, we're looking to explore further and further in our solar system. We're looking to extend human presence beyond low Earth orbit, beyond even the moon. We want to go to Mars, and further. Now, as an aspiring, hopeful, one day in my wildest dreams astronaut, of course I'm imagining all of these exciting adventures that are yet to come for humanity, but I'm also thinking about what we need to do to get there. As we plan to travel further from home, we know that we're going to be embarking on much longer missions, and astronauts will need to live in space for extended periods of time. It's important for us to understand the impacts of long-term spaceflight on the human body as we plan to send astronauts to deep space. Let's talk about one step in this direction, the NASA Twin Study. In this experiment, astronaut Scott Kelly spent 340 days on the ISS, while his twin brother, Mark, remained on Earth. One of the most striking discoveries in this study was the changes in Scott's telomere length. What even are telomeres? Well, they act as protective caps on the end of our chromosomes, preventing the degradation of DNA while our cells replicate. Why do we care? Human telomeres usually shorten over time, which contributes to aging, is associated with major diseases and mortality, and is linked to cancer risk. Okay, got it. Telomeres unfortunately shorten as we age. Now, get this. Scott's telomeres lengthened while he was in space, and then saw a rapid shortening within 48 hours of returning to Earth. This was surprising, as the increased exposure to radiation and the stressors of spaceflight were expected to shorten Scott's telomeres at a faster rate than his brothers. See, the effects of microgravity, radiation, and other factors of spaceflight on telomere dynamics are not yet well understood. So it would be beneficial to isolate each factor in the study to discern which is responsible for the observed phenomenon. This inspired an idea to study the effects of changes in gravity on the genetic regulation of human telomeres. I'm leading a team of students selected to participate in the Canadian Reduced Gravity Experiment Design Challenge. This campaign is in collaboration with the Canadian Space Agency, the National Research Council of Canada, and SEDS-Canada. We're designing, building, and testing an experiment to be flown on board NRC's Falcon 20 on a parabolic flight this August. Flying in simulates microgravity conditions, providing a research experiment that is unparalleled here on Earth. We designed a sealed and automated system to inject cells with lysine solution prior to and after periods of microgravity. The lysine solution will break the cell membrane, which preserves their nucleic acids for post-flight analysis. Then we can measure the expression levels of genes that contribute to telomere extension, degradation, and regulation. We hope to both further the understanding of how changes in gravity affect telomere length during spaceflight and the implications this may have on human health here on Earth. Thanks to my amazing teammates who are all working hard to bring this project to life. Dunia, Anthony, Taylor, Luca, Mackenzie, Miranda, Emma, Nicole, Samantha, Krish, and to our faculty advisor, Dr. Rodrigo Fernandez-Gonzalez. Thank you.
Okay, another great presentation. And actually, as somebody who has a background in biology, I found that your presentation was really interesting, Erin. So thank you so much for that.